of these elements. So they cause residual elements or also oligo elements. And they are very specific. Okay, then when we will talk about proteins, for instance, we can talk about these very residual elements. So in our body or in the living organisms, we can imagine that these elements, they are part of two big families. So the inorganic substance, and for instance, the water. Water, it's around in our body around 65% is water, and then we have other inorganic compounds that are non-soluble, like calcium phosphate, for instance, that is very important for our bone. It's one of the main compounds of our bone. And the other family of substances, do you know what are? So if here we have the inorganic substances, what are the other substances? Do you know? What could be? No one's know. It's very complicated.
We don't see the slideshow.
Really?
Yes.
What do you see? Just
a white slide.
so you can't see nothing.
Yeah.
So can you see or not? What you can see?
We see the file, but not the presentation.
So it's empty. It's like it's empty.
Now we see the files and
Now you see, I think all. So you see the platform of
yes.
eLearn. Yes. Okay. And if I put this file, you can see what you can see now.
I don't see the presentation.
Oh, this is not good.
I see the file of the lecture with all the different documents.
Yeah.
But not the presentation.
And now?
It's just a white screen.
Oh, I don't understand why. Let me see if I put... And for instance, if I put and this, you can see?
No,
No. I'm trying to do something, okay? I don't understand why you can't see it. And do you know how can I share with you documents with Teams? If I send you the...
maybe in the chat of the conversation.
Yeah.
We can send some files through the chat.
Okay. Do you have it? No, not yet.
Yes, I think.
And also, I have a message because it's very strange why I can't see when I put my presentation, I can't see your message, for instance.
Okay.
Can you see my lecture now?
I've downloaded
Yeah.
and I think it works. Yes.
Okay, it's okay.
Yes.
All of you have and I can tell you when you need to move to change the slide. The only thing is that let me see. Apparently, there are some students from CPCM that are waiting for my authorization to come to the discussion.
Yes, I think Bastien doesn't have the invitation or the link to Teams.
But I don't think that it's me because okay. And now it's okay for Bastien?
Ah, yes, yes.
Yes?
He just have the right to enter in the groups, so it's okay.
It's okay for Bastien?
Yes.
Okay. And now I have another. Okay, so let's try again. Sorry. Okay, let me see. So do you have now the presentation? Yes? All of you? I would like to have your confirmation.
Yes, yes.
Yeah?
Mm.
Okay, I'm sorry. It's complicated for me. Okay, now I see. Because actually, when I put my presentation to guide me, okay, I will do another way. I lost your message. I don't know why. Okay, but I put in my other screen. It's okay. Okay. Sorry for that. And if I put like this. So we are in slide 4. Can you put in slide 4, please? It's okay?
Yes.
Okay, so, and now you have everything. So just before, just to see, you see, I put in the slides So just the first slide about the showing you some examples of biogenic elements that are essential for us in the composition of the living organisms, and then I just was telling you that we have two big families. So the inorganic substances like water and other components that are non-soluble as calcium phosphate that is very important in the composition of our bones. And the other substances, of course, are the organic substances. And the organic substances, they include four big families. Do you remember what are these four big families that we talked last time? It's the four. It's the four. Um, it's the four big families of biomolecules. So that includes proteins, nucleic acids, carbohydrates, and lipids. And but as I told you, we will discuss only about in our course. We are just interested for the natural polymers on proteins, nucleic acids, and carbohydrates. So we can move to the other slide. And today, our course oral lecture, it will be we will discuss about the carbohydrates. So, do you know what are carbohydrates? Or are you familiar with this word? What are carbohydrates? When we talk, for instance, if we discuss with a colleague or with a friend that it's not a chemist and that we want to discuss about carbohydrates, what do you think that they? If you ask your colleague or your friend, what are carbohydrates? What what do you think that they what can they tell you? So are we familiar with this word or not? Do you know what are carbohydrates?
They are basically sugars.
Yeah, they are sugars. Very well. And I think that if you put the word, I did the experience of carbohydrates in Google, look what I have. So carbohydrate is associated with food. When we talk about carbohydrates, um, so what we think? We think about food. Carbohydrates, they are the main base or the main components of our food, what we eat. And you see, if you can do the experience, you can put carbohydrates in Google and put image and you will see the same page as I have here. Okay, so carbohydrate is really associated with what we eat about, so with food. So they are the main compounds of fruits, of cereals, etc. So we also discussed a little bit last time about the function of some carbohydrates. And the two main functions are as energy. So now we are in the other slide, sorry. Um, so the main, I forgot that you can't see when I put my slide. So the main functions of carbohydrates is energy source and is a structural material. And of course, that is the starting point for the other organic compounds, as we will see when we will talk about the nucleic acids, for instance. So, as I put here, one of the primary functions of carbohydrates is to provide our body with energy. And as we said just before, it's an essential part of our diet. So we found, of course, in cereals and fruits, but also in vegetables. And carbohydrates serve as a structural material. What is the main if we talk about structural material, we think to cellulose in plants, but we also discussed last time that another polysaccharide that is very important in terms of structure of some animals is chitin. Okay, um, so there are also very interesting another function is that we can with enzymes we can use the enzymes to and to have simple sugars and to do more complex molecules, and it's really very interesting this kind of way to create molecules, okay? And as I said, it's serving as components of nucleic acids and other metabolites. So if we move to the other slide, we have then the chemical structure of a carbohydrate. And if we look for the chemical, this basic chemical structure, what we have carbon, and then what we have. What is the molecule that is the in the right side of the carbon?
Water.
Water. And in this way, it's because of it, it's calls carbo from the carbon, hydrate from water, you see? And in this way, we can all the time to identify a carbohydrate. And here, just before to start with the nomenclature, to learn how to do the nomenclature of a carbohydrate, we have here some examples. So do you know what is the more, or well, for instance, when you talk about carbohydrates, of course, for me, for instance, I think about polysaccharides. When someone talk with me with carbohydrates, I think about polysaccharides. But there are other thing that we should think when we talk about carbohydrates is glucose. Glucose is really the most important example of a carbohydrate. Okay, so we have glucose, we have fructose and sucrose. These are some basic um, carbohydrates, okay? And for instance, for simple carbohydrates, the ratio of carbon, hydrogen, and oxygen is 1 to 1, but we have also other carbohydrates that has other elements. So can we tell me or give me an example of another carbohydrate that it's not only composed by carbon, hydrogen, and oxygen? So here we have the examples of glucose, fructose, and sucrose. But if you see the chemical structure, we only have carbon, hydrogen, and oxygen. But we already discussed in our lectures about other kind of um, um, carbohydrates with other elements. Do you know an example, for instance? Do you remember? Chitin, for instance, or chitosan, okay? We have nitrogen in these molecules. And what is interesting is to know that the presence of this nitrogen will give another function to our polymer. So the structure is the same, but with the presence of these other elements, so the function of this polymer, it's different. It could be different, okay? For instance, you know what is the difference in terms of biological function between chitin and cellulose? So cellulose doesn't have nitrogen and chitin has, and chitosan also. Do you know what is the main difference in terms of biology?
No.
Sorry?
No, I don't know.
Oh, you don't know. Okay. So the presence of amino groups in chitosan, for instance, give to chitosan some antimicrobial properties, and it's the same for chitin. Okay. And for instance, cellulose, if we want that our material, or if cellulose has antimicrobial properties, we need um, to introduce to incorporate this function to cellulose, or by chemical or enzymatical modification, or just, for instance, if we have a material, so we can um, modify the structure of the cellulose by grafting another molecule that has this property, or we can, in our material, final material, we can introduce components or molecules that has this property. Okay, but for instance, with chitin and chitosan, we don't need it because they already have these functions. So if we move to the other slide, here we have in this slide, so 14, we have some rules to know, to identify carbohydrates, and how to, yes, how to identify carbohydrates. So the simple carbohydrates, like glucose, lactose, or dextrose, they end all the time with O's, okay? And these simple carbohydrates, the number of carbons, they can be from 3 to 7 carbohydrate, different sorry, from 3 to 7 carbons. So we have 3 if it's a carbohydrate with 3, like this, 3 um, carbons, so it's 3 DOs. And for instance, the glucose, we have 1, 2, 4, 3, 4, 5, 6. Okay, so it's 8 exos, 6 carbons. And also, depending where is the functional group of the carbohydrate, we can call ketose if it's a ketone, or aldose if it's an aldehyde. But we will see this after. Okay? Oh, sorry. Also, what we can see is saccharide. It means, as Unimaya said, sugar. So we can, it's the same if we use saccharide or sugar, okay? And now we will see the 3 main um, uh, families or groups of carbohydrates: the monosaccharides, the disaccharides, and the polysaccharides, okay? So if we move to the other slide, what do you think that, why it's monosaccharide? What it means, monosaccharides? Mono, it's from one sugar. So we have molecules with one sugar. So the most common, as I said before, it's glucose. And they, as also I said, they are characterized in terms of the number of carbons and the position of their carbonyl group. If the sugar has an aldehyde group, so now we are in the slide 17, if we have an aldehyde group, we call aldose. And if has a ketone group, we call a ketose, okay? And now I have 2 examples. So an example 1, it's the other slide. We have 2 examples. We have, for instance, glucose and fructose. And now, in this example, we will talk about the position of the carbonyl group. So here it's for, oh no, you can't see, sorry, it's terrible. So glucose, if we go through the other slide, so glucose has the carbonyl group at the end of the structure, you see? So it calls an aldose. And if the carbonyl group is in the middle of the chain, the backbone, it means that is ketose. So if we go if we do a click, so we have what we call exose, because it's an aldose with 1, 2, 3, 4, 5, 6 carbons, and we have exose. So exose with a ketose we call a ketoexose. Okay. Then I have some exercises for you. You will see it's very simple. Maybe the people from chemistry, the students from CPCM, you already discussed about this kind of things in organic chemistry. Yes.
About functional group, yes, but not in relation to sugar.
The sugars. Okay. Very well. Okay. Yeah, so what I have here, just for your information, it's, for instance, now we are discussing the monosaccharides, glucose and fructose, and these two molecules, they make what we call sucrose, okay? And fructose is the common sugar that we have in the fruits. Okay, so let's move to the other slide. Now it's the example 2, and okay, here what I want to talk with you, what I would like is, so in this case we have glucose and galactose. So galactose is part of the lactose, it's the milk sugar, okay? And we have the chemical formula, and as you can see, they have the same chemical formula, but they have a difference. Can you see and tell me, please, what is the difference between these two molecules?
Position of the hydroxyl group.
Yes. In which position?
Fourth carbon.
Yeah, exactly. Can you see? If you go if you do click, so you can see that there is a differences in the position in the carbon 4, okay? It's the position of the hydrogen and the hydroxyl group. So it means that they have the same chemical formula, okay? In this case is C6 or 6 carbon, 12 hydrogens and 6 oxygens, and it means that are isomers. I think that you already know this term, and this, as I said, it means that they have the same chemical formula, but they have different different arrangements of the atoms. Okay, and this is important. For instance, here, why this is important? Because just now, until now, I was showing you the carbohydrates in the linear form. But, for instance, when you see the articles that I give you to see polysaccharides on proteins, did you see your molecules in the linear form or in the ring form?
The ring form?
In the ring form, yeah. So when our monosaccharides, they have 5 or 6 carbons, they can exist in the equilibrium between the linear chain and what we call the ring-shaped molecules, okay? And in aqueous solutions, usually they have the form of a ring. So you can see. Oh no, you are sorry. I forgot that. I can't show you. So we have both structure, so linear and ring, and it means that the fact that the intramolecular OH group reacts with the carbonyl group, that it's in the end, in this case, it's in the end of the structure, the formula, okay? And the formation, what we have the formation of a hemi-acetal or a hemi-ketal, resulting from the addition of an alcohol to the aldehyde or a ketone. It will depend what we have, okay? If it's a ketone or an aldehyde. And here we have, for instance, um, the two examples. We will see in the next slide the differences between if we have a monosaccharide with a beta or a alpha position, okay? So here we have the two examples of glucose. If we have if the position is alpha or beta. So glucose in the ring form can have two different arrangements, as I said. So beta or alpha, it will depend if the hydroxyl group is belong to carbon on one in the sugar. It is said to be in the alpha position. If it's above, if the plan is above, so it means that is in the beta position. Can you see here where it's the OH group in the carbon 1? Can you identify carbon 1 in these structures? So you see the oxygen, and the carbon 1 is the carbon that you have in your right hand. You see? And here, if the OH group is above, you see? Can you see? If it's above, yeah?
Yes.
Okay. So you can see. And if it's in the above to the plan, it means that it's a beta position. And of course that it will be our polysaccharides, as we will see in the next lectures, will be very different properties if they have the alpha or the beta position, okay? So in the next slide, we have the stability of the beta form. So in the equilibrium, actually, the beta anomer of the glucose predominates because of the OH group of the anomeric carbon is in the more stable equatorial position, okay? But for alpha glucose, the OH group of the anomeric carbon is axial. And you should remember that for glucose, it's all the time if its alpha is axial, okay? And you can see here in your in this schema that I have. So it's in the equilibrium between the different positions. Ring with alpha ring, okay? The linear chain, and then the other, the circular, the ring, but in the beta position. So in the next slide, we will move to the disaccharides. And here it's D because it's 2. It means that we will have 2 sugar in our molecule, okay? So when it means that there are a reaction that it's happening between 2 monosaccharides, okay? So, and I think that you already talk about what is a dehydration reaction. So the OH of the monosaccharides combine with the hydrogen of another monosaccharide, and what it happens when OH group of the saccharide react with hydrogen of the other saccharide.
Water molecule element.
Yeah, it's the formation of a water molecule, okay? And in this case, for instance, with the example that I gave you before, glucose plus fructose, we have the sucrose formation, okay? And now, finally, we are moving for the polysaccharides. So just we can go another through another slide. And what it means poly, it means many. So we will have a lot of mono saccharides in our molecule, in our macromolecule. Now we can start to talk about macromolecules, and even we can talk about biomacromolecules, because our macromolecules, they are from natural resources, so we can call biomacromolecules or polymer, okay? So it means that we have a long chain of a monosaccharide linked with glycosidic bonds. Oh yes, sorry, I forgot. No, I don't have it here. No, maybe. Yes, I forgot to tell you. If you go to the disaccharide, sorry, if you go to the disaccharide slide, so there are the reaction, and as Unimaya told, we have the formation of water molecule, and this kind of linkage between the two monosaccharides, it calls a glycosidic bond. Okay, this is very, very important. It's glycosidic linkage or bond. Okay, this is really very important too. It's what do the big difference between the different molecules or biomolecules. Okay, so let's move to the polysaccharides again. So regarding the polysaccharides, it's, as I said, a big chain of monosaccharides, and they can be branched or unbranched. So, for instance, can you tell me an example of branched polysaccharide and an unbranched polysaccharide?
Cellulose is an unbranched polysaccharide, I think.
Yeah, good. And another example?
Lignin, maybe.
Sorry?
Lignin or lignin.
Yeah, but now here we are not talking about this kind of molecules.
Okay.
Yeah. We talk about polysaccharides. And lignin, it's not a polysaccharide, okay?
Okay.
But it's a it's a biomacromolecule. That it's very complicated to work with with them. Okay?
Okay.
So, yeah. So you have another example. Is, for instance, the starch. Starch, it's a branched. It's composed by two different kind of polysaccharides, and one of them, so the amylopeptine, it's branched. You can see here in the, so in the left side you have cellulose fibers, a representation of course, and the cellulose structure, and in the right side you have starch, so almidon in French. So you see, starch is composed by amylose and amylopectin, and the amylopectin it's branched polysaccharide. So we consider that starch is a branched polysaccharide. Okay. So if we go now for another slide, so we have an exercise. The first one, it's very simple because we already did this in last lecture. So it's to show you, you see here, so you have, if we still with branched and unbranched, you see here starch in the right, in the left side, and glycogen. Also, you see here, we can see this branched polysaccharide, but cellulose, no, it's a linear polysaccharide, okay? So the question is, what is the role of each of these polysaccharides? Do you remember? Starch, what is the main role of starch?
Storage.
Is store energy, yes, and glycogen. But store energy in plants and glycogen, what is the main role?
In human storage.
It's the same, it's storage energy, but in animals and bacteria. And if you see, so in the figure, if you see below the starch and glycogen and cellulose, we have microscopy images. Can you see? And do you see how it's similar, the morphology of starch and glycogen? You can see it. So it's to store energy. But, and they are branched polysaccharides. And if we see cellulose, that it's a linear polysaccharide, what you can tell me about the morphology of cellulose if we compare with the other two? For instance, no, first, sorry, Alexi, yes?
Yeah, yeah.
Yes.
No, it's like very organized. So with linear chains.
Yeah. So their morphology is fibers, okay? As we talk, okay? So their role in nature is to give structure. So I have now the answers. And here, so if we go two slides more, I have the role, I described the role of starch, glycogen, cellulose, and chitin. So chitin is the same that cellulose, but in animals, in the exoskeleton of the animals, like insects and some crustacea. Okay, so yeah, if we go a slide further, it's the, now I don't have the numbers of the slides. It's the this with that you can see the structure of cellulose, woods, and a crab. You have a forest and a crab as image. You see what is the slide? Yeah, are you in the good slide? Yeah, okay. So I have here just these two structural polysaccharides as example to show you. But this, of course, that we will discuss in detail during the next lectures, because this is very important for the properties of the materials that we want to do with these polymers, but also regarding their properties. And I put here just because I think it's important today for you to stay in your mind the structure of these polysaccharides, okay? And so we have at the beginning the different monosaccharides that link it together to do the polysaccharide, and then these polysaccharides, they will assemble together the long chains to form the nanocrystals, and then the cellulose nanofibers, and then the fibers, okay? And it's the same for the crab, for instance, for the exoskeleton of the crab. So, and what is important is to see that they really have the same structure and almost the same chemical formula. But in the carbon 4, it's not an OH group that we have in chitin, okay? And now let's move to the other slide, and I have some to continue talking about polysaccharides, and I have the first example of your mind mapping. Can you see the first example?
Yes.
It's the slide 35. Yeah? All of you are in this slide?
Yes.
Okay. So, who did this slide, this mind mapping?
To mine eye.
Toma and you, yes. So thank you very much. Don't forget for the other people. So I received 3 until today. Don't forget that you should send me until the end of the week, okay? But it's very funny because we just talk about cellulose, chitin, and starch, and I receive your mind mapping about cellulose nanowhiskers, chitin nanowhiskers, and starch nanowhiskers. So it was really very funny because in this way I can show you the mind mappings that your colleagues they did, and they are very good. I I would like to tell you congratulations because um, they are really better than those that I asked your colleagues last year. And yeah, you put more details, you go through more, yeah. So thank you. They are really very good. So I don't know if Thomas and Alexis, do you like to discuss a little bit about your mind mapping and explain us what you did?
Yes, yes.
Yeah. So go ahead.
So cellulose nanowhiskers are crystallite, have the structure of crystallites of cellulose,
Mm-hmm.
as we can see on the mind mapping, and we have seen before in the course, cellulose have two different crystalline form in plants with alpha glucose and beta glucose.
Mm-hmm.
This type of cellulose nanowhiskers have a rod-like shape that goes to 3 to 30 nanometer wide, and they can self-assemble into curl nematic liquid crystal. Uh, I understand that this type of structure is could be explained with an electromagnetic field, but I'm not sure.
Yeah.
Um, Concerning their properties, they have good optical properties thanks to their anti-reflective behavior, and we can use it to into the transmission of light. Because it's at the nanoscale, they have a high surface to volume ratio that makes this type of particle highly reactive and functionally visible. Moreover, about the surface functionality, we can play with the charge of the surface or the active groups that are present at the surface. Um and yeah, they have a good mechanical properties with an elastic modulus about 140 gigapascal.
Mm-hmm.
We saw that this type of particles are made with acid hydrolysis of cellulose plant, if I remember well, or
Yes.
tunicates. As we can see, the origin comes from plant tree, bacteria, or tunicates.
Mm-hmm.
We've shown that the cellulose nanowhiskers in coming from tunicates expect a larger aspect ratio, so
Yeah.
they're
They are
the
really
rod-like,
very, very long. Yeah.
yeah, compared to the plant one. And we can use it, expect to use it in smart coatings, pharmaceutical, nanocomposites. In the publications, they have made a comparison between carbon nanotube and cellulose nanowhiskers and say that is a good thing unless it have a lower elastic modulus. And we can use it in electronics in regard to the optical properties.
Yeah, good, very good. Okay, so do you want to tell us something else?
No, it's okay. It's okay.
It's okay. Okay, if we go just to the other slide before, please, where when we have the two structures of cellulose and chitin, you see? So you see, so we have the cellulose chains that will assemble together to form. You see in the second we can see cellulose. They call it cellulose nanocrystals. Okay, so actually cellulose is what we call a semi-crystalline polysaccharide or polymer. It means that cellulose has, as we can see here in this figure, crystalline regions and amorphous regions. So, as Alexei tells us, with very strong acid hydrolysis acid reaction, we can dissolve the amorphous regions and we stay just with the crystalline regions of the cellulose, and we have what we call, now if we can go to the other slide to the mind mapping, we have the nano whiskers. Okay, so it's very nice. You put the origin of cellulose, and they are all correct. Some from plants, from trees, bacteria, and also from animals. So it's a marine origin. And you put the structure. So for you, it's very important what you put here regarding the crystallinity. And we have this. We have three main structures. We also have gamma structure, but it's not very common. So the two most common it's alpha and beta, as you put here. It's very funny that you put the same image that we just discussed before. So this is very good. And then you talk also about the properties of these nanocrystals, and you put the three main important properties. So, of course, it's the surface, the possibility, because so these nano whiskers can be used, for instance, for this kind of applications, and we can use as such, so the nano whisker, or we can also to modify this nano whisker by incorporating other molecules. And it's important because this in the form of the nano whisker, we have a lot of the surface functional groups that are available to do some enzymatic or chemical modifications and to incorporate a new function to our cellulose nanowhiskers. The mechanical properties, for sure, it's one of the reasons why we use these cellulose nanowhiskers. And the optical properties, it's very important. For instance, when I started my thesis, it was in 2006. Sometimes it's very very long time ago. It was the moment when we started to work about transparent nanocomposites. My thesis is about transparency, about transparent nanocomposites, and we used a lot of these nano whiskers because it's transmittance. It's transparent, and it was possible at that time. Now it's not new, but at that time it was very new. We published very nice articles about this plastic because we used other kind of matrices from natural polymers with these nano whiskers cellulose to do transparent materials. Okay, So nice. It was nice to have this example to show you an example of a polysaccharide in another form. Okay. So the next slide is Anais. Anais, are you there?
Yes.
Yes. Thank you, Anais, for your very nice figure that you did. Did you like? Do you like to discuss a little bit? It's up to you, okay? If you just would like to tell us what you did and why and how.
Yes, okay, I can talk about it.
Yeah.
So I read an article about the kitten whiskers, and in these articles, I I could I had seen the origin, the applications, the properties, and the structure and the morphology of kiting whiskers. So the origin of kiting whiskers can be from two ways. There is the biosynthetic ways and the non-biosynthetic ways. So for the biosynthetic ways, it can be extracted from living organisms such as shrimp, crab, tortoise, and insects. And for the non-biosynthetic ways, it can be extracted from the polymerization of chitobiose oxazoline derivative.
Mm-hmm.
Concerning the structure, kiting whiskers has three allomorphs, alpha, beta, and gamma forms, which depends on the different resources.
Mm-hmm.
The kiting chains are organized in sheets helped by a number of intersheets hydrogen bonds. Also, the structure of kiting is very analogous to the cellulose.
Yeah.
And
the only difference is in the carbon 4, as we already discussed about it. Yeah.
yes. And the length of kiting whiskers depends on the origin of the kiting.
Very well. Yeah.
And the kiting whiskers are similar to the cellulose whiskers because they have rod-like morphology.
Mm-hmm.
Then kiting whiskers have can bring different properties to materials. Like for example, biocompatibility, biodegradability, high surface area, nano-size dimension, and absorption properties. Then concerning the screen pen beta kitin nanofibers, it has a highest shear stress and viscosity due to a high aspect ratio.
Mm-hmm.
After for the
Just sorry, Anais, just to tell you that when we will discuss more in detail about kiting, I will show you some images of microscopy to show you the different morphologies of alpha and beta chitin, and the same for cellulose. Okay, you can continue, Anais. Thank you.
then it can bring the thermal stability and oxygen permeability. Also in non-aqueous solutions, in non-aqueous solvents, it has a good dispersion and associated to polymer, it can bring the reinforcing effects depending on the aspect ratio of nanocrystals and also on the method of sample preparation. In the articles, they showed that kiting whiskers that were prepared polymers with associated with kiting whiskers that were prepared by casting and evaporation methods were more efficient than those prepared by freeze drying and hot pressing methods.
Yeah, it's true.
And also it can bring it can bring barrier properties on the surface of material.
Yeah, and in your paper, they talk about the different barrier properties. Do you know what kind of barrier properties it could be?
There were hydrophobic properties, I think.
Actually, when we talk about barrier properties in a material, we can talk about three main kind of barrier properties. So it's a barrier against microorganisms, barrier for the water or water vapor, and barrier to gases. You understand? When we put,
Yes.
yeah, actually it's these three things. The microorganisms, because when we, for instance, if you do a membrane with this kind of materials, it's important. For it depends of the application, but maybe you want a material that has a barrier property against the microorganisms or against water. You don't want that water or vapor water past your material or other solvents or gases. It's also important. Okay, oxygen or yeah. Okay.
Yes.
So when you put here that barrier properties, it's barrier, it's part of the properties of these chitin whiskers. It's true, but you need to mention what you mean for barrier properties. Just put water vapor, gases, and microorganisms, for instance, because in this way it's clear for the other two persons. Okay.
Okay. And finally, the applications of these chitin whiskers are applied in different domains, such as in biomedical materials, like for example with hydrogels and scaffolds. Also in agriculture, in water treatment, in cosmetics, tissue engineering materials, and food industry, and also drug delivery. They can also be used as raw materials with the chitosan nanoscaffolds, and they are used as to reinforce polymer nanocomposites.
Yeah, like cellulose. Very good, Anais. Yeah, thank you. And the next one is Oriane. Yes. Oriane, are you there?
Yes, I'm there.
Yeah. Do you like to discuss a little bit your mind mapping?
Yes. Okay. So, starch crystals in plants. So, we can see in this review they use starch from corn, wax maize, pea, and potatoes. They have starch have a circular structure with crystalline and amorphous parts.
Mm-hmm.
So you can see that the layer of one layer of crystalline, one layer of amorphous part. And in the crystalline part, there is a nanocrystalline starch. So they're like lamellae.
Yeah.
And these nanocrystal are most of the time chemically modified by chemically or by another strategy like graph or their graph.
Yeah, graph to or graft from. Yeah.
Yeah. To improve the interfacial adhesion with other material, like matrix, to make full full degradable nanocomposites. Some companies make charge in tar to I don't have the word. After
Take your time.
Okay.
No, don't worry. Take your time. It's difficult for all of us. It's something new that we are doing, so it's very strange, also for me. So take your time. Don't worry. Okay.
Okay. So they use nanocrystals in tar to replace black carbon. This is the objective. Because of the big properties, good properties, we have a good percolation mechanism thanks to the bond. So we have good thermal properties, barrier properties. I think that is for microorganisms. There is a good solvent absorption and good, very good mechanical properties in tensile tests, for example. So there is also a very strong interfacial interaction that is very interesting also for the mechanical properties.
Okay, very good. So just one comment about the barrier properties. So you tell us that maybe it's for microorganisms. Pay attention because starch doesn't have antimicrobial properties. So it could be, but in this case, we should we should to know, for instance, if it's a membrane, the pores of the membrane should be smaller than the microorganism, because otherwise the microorganism can pass through the pores of the material. Okay? So it depends, because for instance, the starch could be a barrier for a microorganism. In this sense, if the pores of the material of for instance, if it's something for medical applications, if it's for so for surface, for instance, if we want to use this material, so for microorganisms we should be take care, just because it's not anti fungal or anti bacteria. So what we can use is to put, okay, if the pores are smaller than the microorganism have some difficulty to pass through, but it's not killing the microorganism. And for instance, if it's with chitosan or chitin, it could or kill or to immobilize the microorganism. Okay, it's we need to just to see. So I think that with starch, it's more for gases and water vapor barriers. Okay, to control the gases and water vapor. Okay, so thank you to all of you to be here today in these conditions quite strange, but we need to. So as I tell you, I would like to talk about with you today about the project, but it's not. I need to think to reformulate the next lectures, and to do, I don't know, I need to think about it, even the conditions. We also didn't have time to do the exercises. What I will do, I will put everything in eLearn until the end of the week, and also a document to organize the project. So what I suggest is that you share between you this document, So because what I'm thinking that the project will be the presentations, and the presentation could be, for instance, to talk about one polymer, or to talk about some case studies of these polymers in research that we are doing today. Okay. And I will put everything in this document, the different possibilities, and you need to choose what you would like to do in the project. Okay. So next week, I think that we don't have lecture, so you have time this week to discuss and to see what you would like to do, and next week to prepare something if you should present something in 3 weeks. Anyway, in the first I will do myself. I will continue actually what I am doing today. So next time we'll discuss about proteins and nucleic acids. Okay. Okay. So thank you again. And I will put everything in eLearn, and don't forget to send me your mind mapping. Okay. So for me, it's you don't have any question for me because the lecture today is finished. If you have, please go through. It's okay for you?
Yes.
Yeah. And of course, you can send me emails or with questions. You can. I think that I have my phone number in my signature of my email. You can also to phone me, and it's okay. Okay.
Okay.
Okay. See you. Thank you.
Thank you. Bye.
Bye bye.
Thank you.
Thank you. Bye bye. See you later. And take care.
Take care.